Okay, so
Yes, sir.
this will be our last. Okay, so yun nga, importante na you know the types of genomes. It can be double-stranded DNA, single-stranded DNA. So RNA can be double-stranded or single-stranded. So sa single-stranded, it can be positive sense or negative sense. So um, so the process usually we learn from biochemistry. So normal pathway para gumawa ng protein, you have the DNA opening up, tapos from the unzipping of the DNA, you can create the messenger RNA. Tapos yung messenger RNA lalabas sa nucleus, pupunta sa cytoplasm para kabitan ng ribosome to make the protein. Okay? So yun yung central dogma ng biochemistry. Pero yun nga, may mga ibang dahil sa virus, no, there are possible na ibang pathway like in the case of retroviruses, yung RNA ng retrovirus gagawa siya ng viral DNA tapos may insert dun sa DNA ng host. So hindi hindi usual sa central dogma. So tawag dun ay reverse transcription. So the nature of the genome determines how the genome is replicated. So it depends on what the genetic material is. So pag single-stranded DNA, okay, so let's just for the sake of ito na lang para maganda yung itsura. So let's say you have ay kailangan medyo ano wavy. Ayan. So yan. So you have okay, so you have single-stranded DNA. Tapos, so sa template niya, no? So based on the base pairs, pinag-aralan natin sa biochemistry, gagawa siya ng complementary base. Okay, so magkakaroon ka na the other strand will be made within the cell. So gamit yung host cell, gamit yung transfer RNA ng host cell, gagawa no double-stranded DNA. So now you have the double-stranded DNA there. Okay? So this can be replicated. So gamit itong double-stranded DNA, so gagawa ka ng messenger RNA based on the uh, dun sa uh, dun sa isang strand, no? So ibang mangyayari pag single-stranded RNA. So burahin natin 'to. So if a virus has a positive sense na single-stranded RNA genome, it can be translated directly. So halimbawa ito ay single-stranded na RNA, no? So pwede nang kumapit diyan yung mga ribosomes, no? So lalo yung mga ribosomes, so you have the small and the large ones. So the large and the small. So papasok diyan yung RNA, tapos gagawa siya ng mismong eventually you will form the protein, di ba? So dahil sa messenger RNA, you will form the protein na kailangan nung uh, yung kailangan ng virus or ng virion. And then, yung genetic material niya nagre-replicate dahil nga gagamitin niya yung host cell para mag-replicate yung uh, single-stranded RNA. So that's for positive uh, SS or single-stranded RNA. So directly gagawa siya ng mga proteins. Pero hindi possible yan for negative. So yung negative,
Listen.
iba yung genetic code niya. So yung positive, iba yung genetic code niya. Di ba nagko-complementary base pairs yan? Halimbawa dun sa kabila, meron kang yun. Diba, meron ka lang G, G, G. So, dun sa kabila, sa kabilang strand, sorry, ibahin ko yung drawing. So, ito yung single-stranded na positive, negative sense, no? So, hindi siya pwede kabitan ng ribosomes, no? So, kailangan niya munang gagawa ng complementary strand. So, yung kaninang positive sense, no? So, dun sa kabilang sense, so dun sa kabila, so meron kang, teka lang, hirap mag-drawing. So dun sa kabila, so yung positive sense mo, gagawa ng replicate nung mga base pairs dyan, yung bases mo dyan. So, lalagay ka ng so dun sa kabila, no? So, ito yung negative sense, tapos ito yung positive. Okay? So, bale, magre-replicate siya muna, tapos, pag nagawa na yung complementary strand, so ayun, nawala. Ito ng positive sense na nagawa mo from the negative, yun na yung magiging malalagyan na ng ribosomes na pwede nang gawan ng proteins. So dito na yung production ng genetic material and protein material ng new virion. Okay? So, a virus contains a negative ssRNA genome, cannot translate it until yung negative single-stranded RNA is replicated into the positive single-stranded RNA. So, kasama yan, merong kasama yung virus na RNA polymerase para magkaroon ng positive na single-stranded RNA. Okay, so now you can form the virion particles. So ito yung process, no? So hindi ka basta-basta pwedeng gumawa ng single-stranded. Pag from negative single-stranded RNA, kailangan mo muna yung RNA polymerase para gumawa nitong positive single-stranded RNA. Para dumami siya, so gagamit ka ng RNA polymerase. So para mag-replicate pa, dumami pa yung virus. So based on the positive single-stranded RNA, you make more of the negative sense single-stranded RNA para mag-produce ka ng mas marami. And then the cycle repeats itself. So gagawa ka ng positive na single-stranded RNA, and this is the basis for making viral proteins. So unique ang retrovirus because this will go against the central dogma. So what will happen sa single-stranded RNA? May kasama siyang reverse transcriptase. So gagawa siya ng viral DNA, tapos ipapasok niya dun sa host DNA. Okay? So yung single-stranded RNA gagawa muna ng complementary base pairs na pwede lang gawin for the DNA. So try natin mag-annotate ulit. So let's say we have the single-stranded RNA. Okay? So let's say you have ano pang pwede? U, halimbawa U. Okay, U, C, G. Okay? So meron kang single-stranded single-stranded RNA. Tapos gagawa siya ng complementary na DNA lang, no? So pag DNA, wala kang uracil, meron kang thymine, pero pwede mag-pair pa rin dun sa uracil yung complementary base pair. So dun sa single-stranded RNA mo, so form ka dyan, example lang to So ano ba yung complementary dyan? So from U, meron kang A, tapos yung U, A, tapos pag yan G, tapos C. Okay? Paano kung may paano kung ah, meron kang A sa kabila, no? So halimbawa kung may A ka din sa kabila, so sa DNA, wala kang uracil, you have thymine. So ganun mangyayari. So ang mangyari, itong single-stranded DNA, so mawawala na tong once na makreate mo tong single-stranded DNA, then you form the other strand. So dun sa kabila, gagawa ka na naman ng complementary strand dun sa single-stranded DNA mo. So ano ba yung complementary dyan? So you have, okay, yan, C and G. So meron ka ng double-stranded DNA. Okay? So syempre itsura niyan dapat ganito, no? Ano ba yun? So dapat helix. Tapos yung isa mo, ano ba itsura? Ano pong kulay nun ginamit ko? So yun yung isa. Ay, ibang kulay. Anyway, so yun na yan. Magiging viral DNA siya. Tapos, kung meron kang host DNA, so ibahin natin yung kulay. No? So may host DNA ka. So papasok itong viral DNA. So yun, na-insert siya. So ang tawag dyan sa portion na yan, ang tawag dyan ay yung provirus. So yan, continuous pa rin yung DNA mo. Na-insert lang yung portion. Itong portion na to, tinatawag na provirus. Okay? So ito yung HIV, yung AIDS virus. So tingnan natin in detail ano nangyari sa HIV. So unang-una, so the HIV will fuse with the host cell surface, no? So eventually, usually ito yung mga uh, white blood cells yung immune system, yung sa immune system natin. Okay, so this will fuse and eventually yung mga materials ng HIV lalabas na. You have the reverse transcriptase, integrase, so kasama yan sa pre-integration complex. So eventually, yung viral RNA mo and your reverse transcriptase will form the single-stranded DNA. Okay? So once na ma-form yon, then yung complementary strand ng gagawin. So once ma-form mo yung viral DNA, this is transported to the nucleus and then with the integrase, ma-insert na yung sa host cell. So this becomes the provirus. And then eventually, uh, you have the new viral RNA produced by the host cell mismo, by the DNA of the host cell. And you make viral proteins. So eventually, pupunta sa surface. So you have a new immature HIV virus with all the materials. So as soon as it matures, the protease will release the protein and you form the mature virion. So yun yung cycle ng HIV. Okay, so yung persistent infection, this occurs when a virus is not completely cleared from the system. So hindi natanggal yung virus. So the virus is still there. So it stays in certain tissues or organs. So there are two types, yung latent and chronic. So pag latent, so nandun lang yung virus, walang symptoms. So it lays dormant in the cell. So the virus capable of latency may initially cause an acute infection. So may symptoms sa start. Pero once na matapos yung initial infection, it will stay dormant. So example niyan yung sa herpes simplex virus, no? So yung some examples diyan yung sa simplex virus, herpes simplex virus, you have the varicella zoster. So pag bata, chickenpox. And then pag lumaki, pag adult na, shingles, mas masakit na yung mga blisters na yan. So it's the same virus. You have also the Epstein-Barr virus, which cause mononucleosis. Uh, Studies have shown na 90% of uh, of adults have this. No, hindi naman siya fatal, no. So it just cause uh, production ng mga maraming uh, white blood cells. Okay, so hindi naman fatal. So, related siya dun sa herpes virus. So, ito example ng varicella zoster virus. So, the virus that causes chickenpox. So, pag bata, may maano lang siya, itchy lang yung blisters na yan. Pero pag tumanda, it becomes painful. So, gawa siya sa double-stranded DNA. So, ayun. So, may period with latency, so the virus can reactivate in the form of shingles. So, it's a painful, localized rash on one side of the body. Okay, so yun. So, a chronic infection is a disease with symptoms that are recurrent or persistent over time. So, example yun is hepatitis. So, some viral infections can be chronic if the body is unable to eliminate the virus. So yun, you have a long-term chronic infection caused by hepatitis C. Another example is HIV, no? So there will be a period of chronic infection after a long period of latency. So kaya dangerous yung HIV, no? May time na asymptomatic yung person, walang symptoms. So that will take several years, and then ayun, pag naging full-blown AIDS na, uh, chronic na yung infection. Okay, so yun yung example nung herpes. You have simplex virus. It causes the cold sores. Varicella virus. This causes chickenpox and shingles. Lymphocytovirus. So this causes mononucleosis. And yung Kaposi sarcoma, it's caused by radinovirus. Additional virus, you have yung mga nagkakos ng warts, yung papillomas, and then you have the tumors and vacuolation. So this is uh, in the family of the Papovaviridae virus. So double-stranded yung DNA and negative sense and non-envelope. So under this family, you have the alpha virus and the rubivirus. You have the rubella caused by rubivirus, alpha virus transmitted by arthropods. Under Picornaviridae, you have the enterovirus. So it's a poliovirus and Coxsackie virus. Yung rhinovirus, it's what causes the common cold. You also have hepatitis A. Okay, so yung retrovirus so yung family na ng retroviridae. So single-stranded single-stranded RNA, so plus sense and its envelope. So I've explained in detail na yung cycle ng infection. So yung reverse transcriptase, gagawa siya ng DNA from the viral genome and then ma-insert yung viral DNA dun sa chromosome. So magiging part na no host DNA. Of course, I could not discuss I need to discuss also what COVID-19 yung COVID-19 sickness caused by the SARS-CoV-2. So yung particular name ng virus SARS-CoV-2. So it's family of coronavirus. So it can cause illness in humans, animals, and bats, camels, and civets. So kasama sa coronavirus yung nag-cause ng Middle Eastern respiratory syndrome. So yung MERS, okay? So yung ano na yon, yung sickness na yon, napaka-fatal pero mabilis lang yung transmission, mabilis lang yung progression disease. So hindi masyado nag-spread sa Middle East lang yung uh, kumalat yung MERS-CoV. Pero coronavirus rin yun. Okay? So yan, yung COVID-19, sabi nga may intermediate uh, organism bago mag-transmit sa humans. So sa MERS, ang theory is galing muna sa bat tapos na-transmit sa camel. Tapos eventually na-transmit sa humans. Tapos dito sa SARS-CoV-2, SARS-CoV-1 o SARS-CoV, uh, meron kang wild animal na intermediate. So eventually nagkaroon yung humans. So ang theory dito sa COVID-19, so intermediate host nito, I think it's the armadillo. So yun yung theory ng ibang scientist. So yan yung isang theory. So ganun yung structure ng coronavirus, no? So you have the RNA material. So you have the spike proteins na yung basis ng pag-attack ng antibodies. So you have the lipid layer. Okay. So let's discuss detection of the virus. Okay. So yung cytopathic effects. Cytopathic effects muna, no? So yung sa cytopathic effects, from the term itself, it discusses the cell and what happens or the pathogens that can cause uh, deformation in the cell, no? So yun, makita dun sa cell through microscope, either you have the complete cell lysis, so may rupture ng cell, or merong inclusion bodies in the nucleus or cytoplasm. You can have fusion of cytoplasmic membranes and the formation of multinucleated syncytia. You can have vacuoles in the cytoplasm, shrinkage of the nucleus, changes in cell shape from flat to round, or loss of adherence to the surface of the container. So these are all possible cytopathic effects that can be observed through the microscope. Okay? So hindi siya definitive uh, conclusion na it is caused by a certain virus kasi many virus can cause that cytopathic effect. Okay? So that's one way lang para uh, uh, para evidence that it's caused by a certain virus. Another way is through the hemagglutination assay. So this is a serological assay used to detect the presence of certain viruses in the patient's serum. So from the term hemagglutination, so you have the clumping together of erythrocytes. So yan yung red blood cells. So pag may virus, because of their surface proteins, di ba? Yung H1 protein na diniscuss natin yung H. Basically, they can bind to the receptors of the membranes of the red blood cells, and it causes them to clump together. So that's caused by the virus. So this is observable and without the use of the microscope. So makita mo siya using a well. So pakita ko sa inyo ang itsura niya, no? But this method does not always differentiate between infectious and non-infectious viral particles. So possible kasi kung non-infectious yung viral particles, they can also help uh, clump together the red blood cells. Okay, so hindi siya definitive, but it is one way. Okay, so normally, yung red blood cells mo, magsisync siya sa bottom ng isang container, no? So, so pakita ko lang ang hindi ko ito play yung record kasi baka maano ko ng YouTube. I-upload ko kasi ito eh. So I'm not sure kung baka pagbawalan ako ng YouTube. So I'll just show you um so usually magfo-fall to the bottom yung red blood cells sa isang container, no? So ito magfo-form mong dahil sa gravity, di ba? So kung ganito yung shape ng container, they will eventually settle at the bottom pag walang sakit, pag walang virus, no? So it will eventually sink to the bottom. So pag may virus, so virus binding induces lattice formation. So under more normal conditions, no, magse-settle yung red blood cells sa bottom. Pag meron kang virus, it will cause yung ano, so magkakaroon ng lattice. So yung buong container mo, red. So makita mo from top view dahil nga nagkaroon ng clumping together, no, with the hemoglobin. Okay? So yun yung magkakos ng lattice. So pag meron kang antibodies, so basically, pag may antibodies ka, specific dun sa virus mo, then ang mangyayari is magkakaroon ng red spot dun sa gitna. So kung specific siya sa antibodies dun sa virus, then you will also form that red spot in the middle of the well. Okay? So kung specific siya sa antibodies. Okay? So yun yung gagawin dito sa lab. So meron kang maraming trials or maraming samples, so para makita with assurance at tapos different concentration nung titer. Okay? So ayan yung gagawin. Ito yung makita mo without the microscope. So ma-observe mo yung ano, yung mga red spot kung dahil sa uh, dahil sa ano antibodies from the reaction of the antibodies with the virus. Okay? Okay, so and then you have yung pinaka-definitive or the gold standard is the nucleic acid amplification test. So dito gagamit ka mismo ng pinag-aralan natin sa biochemistry. We're going to use the PCR. or the polymerase chain reaction. So ang role lang nito is to make many copies of the DNA. So kung ang genetic material mo sa virus ay DNA, walang problema. So directly yung DNA kokopihin niya, paparamihin niya. So 20 cycles, that will be a million pieces. And then kukumpara yung sequence ng bases dun sa database nung ano kung anong genomic material nung mismong virus. So definitive to. So gold standard to. Okay, we're about to end. So I'm sorry we have to extend a little bit. Konti na lang to. 10 slides left.